So we've been talking a lot about cytochrome C oxidase, and we've mentioned some really important implications of this enzyme. But I think actually looking at the mechanism of this enzyme is going to help you get a little bit more of an intuitive sense of what's going on. But before we actually delve into the inorganic/organic mechanism, I want to just have you remember several things. Number one, this enzyme is going to pump 8 protons from the matrix of the mitochondria, and it's going to use 8 protons in its mechanism that it's going to get from the matrix. Okay, and just remember those protons actually exist as hydrated protons, so as H3O+ or hydronium. And then it's going to pump 4 protons into the inner membrane space of the mitochondria, and that's going to help create the proton gradient that's required to fuel the mechanical energy needs of ATP synthase. Well, the question I want to have you think of is what's happening to those other 4 protons? Because if we subtract off the 4 that are pumped into the inner membrane space, we're left with an excess of 4 protons. Well, the answer to what happens to those is they get incorporated into molecular oxygen. So if you have oxygen plus 4 protons plus 4 electrons, well, what do you get? Well, you get H2O. In fact, you get 2 of them. Okay, so the net reaction of cytochrome C oxidase is it's going to take molecular oxygen, 4 protons that it got from the original 8 that were pumped from the matrix, and it's going to get 4 electrons that are going to come from cytochrome C that came from cytochrome C ubiquinol oxidoreductase complex 3, and then you're going to get out of it 2 water molecules. And just so you can kind of keep track of it, molecular oxygen, or dioxygen, has 2 atoms of oxygen, so those 2 atoms are taken care of by these 2 water molecules, because each water molecule has 1 atom of oxygen. Okay, so just keep all this business in mind as we're going throughout the mechanism, and I'll point out some important things as we go. Okay, so basically we're going to start off the mechanism with this guy right here, which is called dioxygen. So this molecule right here that I'm going to circle in yellow, this is called dioxygen, or molecular oxygen. Okay. We also have a heme A3. So this guy right here, this is heme A3. So this is heme A3. There's also some other things around cytochrome C oxidase that are important. They're closer to the inner membrane space side. You have a copper A complex. You also have something that's referred to as heme A, and basically the flow of electrons in this system are going to be from copper A to heme A3, and then perhaps to the copper 1+. Okay, in most cases that transfer will be to it when it's in the 2+ state. Okay, we also have this copper plus, and eventually it'll be copper 2+, that's coordinated by this histidine residue right here. There's also a post-translational modification where this tyrosine residue is in a covalent interaction with this histidine right here. Okay, and then some water molecules like to hang out around this histidine residue that's coordinated to the copper. Okay, so basically here's what you have to imagine initially. There's an orbital around here, right? There's some orbital with the iron and the multiple orbitals, and there's about 6 electrons in there, right? So the very first step of this mechanism is going to be nucleophilic attack on the dioxygen species. Okay, so here's what you sort of have to imagine. One of these bonds, of course, represents 2 electrons, right? So when the nucleophilic attack occurs by the iron, okay, it's going to attack this oxygen. That's a 6 electron transfer. As strange as it sounds, it's a 6 electron transfer, and that's going to force these 2 electrons to come back onto the iron. So in terms of net electron transfer there, it's only 4 electrons. Okay, then what's going to happen is copper is going to donate 1 electron to this oxygen of the phenol ring of that tyrosine residue. Okay, and that's going to force hydride ejection and nucleophilic attack on that oxygen species right there, and the remaining hydroxide ends up with it ends up with an electrostatic interaction with that copper, which will eventually be in the 2+ state as denoted right there. Okay, so mechanistically that's the first step. A little complicated, but I think you'll find the rest of the steps are actually quite simple. Notice that the iron is in the 4+ state for heme A3. The copper B is now in the 2+ state, and it's electrostatically interacting with a hydroxide now. Number one, we've also generated an oxide right here. We've generated an oxide, an O2-, and we've also generated a tyrosyl radical, so that's important. This structure right here, this is called a tyrosyl radical. Okay. Now what's going to happen is we're going to get our first proton that gets pumped in. So here's what you sort of have to imagine here, okay, in my pictures, okay? The top half of this, okay, all this business up here, this is sort of on the side of the inner membrane space, okay? What's down here, this is sort of on the matrix side of cytochrome C oxidase. Remember, it's a protein or an enzyme that's in the inner membrane of the mitochondria. So what's on the top is the inner membrane space. What's on the bottom is the side that flanks the matrix. So there's a hydronium that's sitting right here, and this is essentially the first hydronium that gets pumped from the matrix side. Okay, so here's essentially what happens. One of these water molecules right here deprotonates this critical histidine residue, and number one, that generates hydronium number 1, and this is hydronium 1 on the inner membrane space side. Okay, so that's our first one there. Then what's going to happen is you're going to get this sort of series of proton transfers where this hydroxide that is coordinated to the copper B is going to do a series of proton transfers right here. And it's just going to go on and on. You'll see a whole bunch of these as we go throughout this mechanism. And you can imagine that there's going to be a proton transfer all the way over here until finally you deprotonate here. This deprotonates this aspartate, and then this deprotonates the hydronium. Okay, so the net effect that we get here is that a hydronium's proton ends up getting deprotonated on the matrix side. The proton will ultimately make its way into the bulk of the enzyme, and it will end up getting attached to this hydroxide that's sort of coordinated here to the copper B2+. Okay, and in the process, a water molecule here, which is held pretty tightly by the enzyme, deprotonates this critical histidine residue in the active site, and that's the proton that ends up being created on the inner membrane space side. And this hydronium right here that was pumped from the matrix side, okay, this is the first one that we get on the matrix side. Okay, so we're going to be keeping track of these protons as we go throughout the mechanism. That gets us to the products of Mechanistic Step 2, which are going to be the reagents of Mechanistic Step 3. So now at this point we have our first hydronium that got pumped into the inner membrane space, and now what we're going to do is we're going to regenerate the protonated state of this particular histidine right here that's necessary to get that proton into the inner membrane space. Okay, but to do that, we're going to have an electron transfer. Okay, so here's the idea. An electron is going to come in from cytochrome C, and specifically this is ferrocytochrome C because it's in the reduced state. And then when it gets transferred to copper A, it's going to get reoxidized into ferrocytochrome C. So the electron's going to first come into copper A, then it's going to bounce around to heme A, and then from heme A, it's sort of going to come over here to heme A3, and then it's going to come to copper B, and then it's going to go onto this tyrosyl radical right here, and that's going to basically generate the tyrosine anion. Okay, so the tyrosine anion is shown right here. But there's also something else that's going to happen. So this histidine is going to deprotonate this water, which deprotonates this arginine, which deprotonates this tryptophan right here, and then you're sort of going to get a similar thing that what we saw before. So this water gets deprotonated, This one gets deprotonated, this one gets deprotonated, that deprotonates a glutamic acid, which deprotonates a water, which deprotonates another water, till you finally get a series of proton transfers, deprotonation of water, deprotonation of aspartic acid, and then we get deprotonation of another hydronium. So this hydronium right here, this is number 2. This is the second hydronium that got pumped from the matrix side. Okay. In this mechanistic step, we didn't get one pumped into the inner membrane space. Okay, all we did is we took a proton from the matrix and we ultimately got that proton essentially onto this histidine right here in the active site. So notice that this histidine now is in the protonated state, and we also have this tyrosine. We have this tyrosine anion. The next mechanistic step is actually pretty simple. This tyrosine anion is then going to deprotonate the water that's in a coordinate covalent bond to the copper B here that's in the active site. Okay, and that's going to generate a hydroxide that's in a coordinate covalent bond to the copper B, and it's going to generate tyrosine in the protonated state. So notice here, this is tyrosine as you would find it at physiological pH. And so we're good with that. The next step of what's going to happen is we're going to get water right here that's going to deprotonate this histidine residue in the active site. So water deprotonates that histidine, and we basically generate another hydronium that got pumped into the inner membrane space. So this is the second hydronium, number 2, that ultimately got pumped into the inner membrane space. From there, then what's going to happen is this hydroxide that's in a coordinate covalent bond to the copper B is going to essentially do a proton transfer that we're used to seeing. So this deprotonates that, this deprotonates that. This water deprotonates the glutamic acid, that deprotonates water, and it's just going to keep going all around here through these serines and waters. Deprotonate this water, which deprotonates the aspartic acid, and then that deprotonates the hydronium. So this right here, this is the third hydronium that we pumped from the matrix. So again, we're keeping track of those. So essentially this proton right here that came from the hydronium, essentially what happens is it ends up on this hydroxide that's part of the copper B complex right here. And of course what that generates is it generates histidine in the deprotonated state as shown right here. So histidine is now in the deprotonated state, and now on to mechanistic step 6. So now what's going to happen is we're going to get a series of electron transfers. Okay, so cytochrome C, or we'll call it in this state, it's ferrocytochrome C, is going to transfer an electron to copper A, which transfers it to heme A. Scrolling back a little bit, then that goes to heme A3, and then that's going to go to copper B. And so what we're going to get at the end of this mechanistic step is copper B. Notice how copper B is now in the +1 oxidation state. It was in the +2 oxidation state, but since the electron came in from cytochrome C, it's now in the +1 oxidation state. And something else is going to happen that's interesting. Number 1, histidine is going to do a proton transfer with this water, which will do a proton transfer with this arginine, which does a proton transfer with this histidine. And the story is the same from here. What's going to happen? Let me kind of scroll down. So this tryptophan is going to do a proton transfer with this water, which does a proton transfer with glutamic acid and water and so forth. And you will keep doing the same thing that we did before. We'll eventually get proton transfer with this water, which deprotonates the aspartic acid, which deprotonates the hydronium. Okay, so if we're keeping track of the hydroniums, this is the fourth one that's gotten pumped from the matrix. Okay, so now what we've done is we've regenerated histidine in the protonated state, and now it's eventually going to be ready to do another proton transfer. Now, a relatively simple mechanistic step, copper B is going to transfer an electron to this iron of the heme, and then what's going to happen is oxide is going to deprotonate this water that's coordinate covalently bound to the copper B. And so what you get at the end of that mechanistic step is copper B in the 2+ oxidation state with a bound hydroxide. and this iron 3 plus of the heme A3, which is also bound to hydroxide. So now let's look at the next step, which is step 8. Now here's what's going to happen. Water, once again, is going to deprotonate this protonated histidine in the active site, and that generates our third hydronium that gets pumped into the inner membrane space. So that's the third one, okay, and that will, of course, decrease the pH in there. Now what's going to happen is something that we're, pretty used to seeing. So this hydroxide that's coordinate covalently bound to the iron is going to do a proton transfer with this water, which does one with this water, which does one with that water, one with the glutamic acid, one with this water, and it's going to go through this series of proton transfers until it finally deprotonates this water, which deprotonates the aspartic acid, which deprotonates the hydronium here. Okay, so this is mechanistic step 8. So this is the 5th hydronium that was pumped from the matrix. Okay, so this hydronium was pumped from the matrix, and so we're over halfway done with the mechanism at this point. Now what's going to happen is we're going to get another electron transfer from another cytochrome C, which is going to reduce copper A, which then transfers the electrons to heme A, which will then transfer its electrons to heme A3, which ends up reducing this ferric iron 3 plus into the ferrous state right here. Okay, with still this water molecule hanging out around the iron 2 plus at this point. Then what's going to happen is you're going to get a series of proton transfers just like we've seen before. Let me scroll down a little bit. So this histidine deprotonates a water, which deprotonates an arginine, which deprotonates a tryptophan. And we can keep going around here. Then that deprotonates this water, which deprotonates glutamic acid, which deprotonates a water, and another water, and it just keeps going around here till finally we deprotonate another water, which deprotonates an aspartic acid, which deprotonates a hydronium here that's held in the active site. So that's another thing. This is hydronium number 6 that was pumped in from the matrix. Okay, so that's the 6th one. So that was mechanistic step number 9. And so now at this point, we're going to look at mechanistic step number 10. So in mechanistic step number 10, number 1, a water here in the active site is going to deprotonate this critical histidine residue that regenerates histidine in the deprotonated state. And that's going to generate our last hydronium, number 4, that was pumped into the inner membrane space. So if you go back and look, here's the third one, here's the second one, and here's the first one over here. So in total, we now have all 4 protons, or 4 hydroniums, that ultimately got pumped into the inner membrane space. But we're not done pumping protons into the enzyme itself through the matrix of the mitochondria. And to do that, we're going to have to look at this mechanistic step right here. So there's also another series of amino acid residues and waters that exist in the active site that's sort of separate from these guys over here. And these are the amino acid residues and waters that are over here. Okay. There's also another hydronium that sort of terminates that tail. And so what's going to happen is this hydroxide right here is going to deprotonate this tyrosine residue that we have in the protonated state, which deprotonates this OH group that's attached to some R group right here, which deprotonates a water, which deprotonates a threonine, which deprotonates a lysine, which deprotonates another water, which deprotonates a serine, and then finally that's going to deprotonate a hydronium. So that's another thing. This right here is which mechanistic step? It's number 10. So that means this is hydronium number 7. That's basically pumped in from the matrix. Okay? And that leads us to the final mechanistic step of cytochrome C oxidase. So hopefully this will make a little bit of sense. Okay, so what's going to happen now is we're going to get our final electron transfer. So cytochrome C, the final cytochrome C, we could even call this cytochrome C number 4, even though it's not what it's called. It's the same cytochrome C as before, but it's the last one. It's going to transfer an electron to copper A, which transfers to heme A, which will then transfer to heme A3. And then finally, that electron will go to this copper 2+, this is copper B, right? And that reduces it into the resting state of the copper B, which is in the 1+ oxidation state. And just keep in mind, we also have the iron in the 2+ state, which is how we started the enzyme. And then the final thing that's going to happen sort of around that same time is this histidine will deprotonate a water, which will deprotonate an arginine, which will deprotonate a tryptophan. And the same thing that we're used to seeing so often. The tryptophan will deprotonate. Let me scroll down a little bit. It'll deprotonate this water right here, which deprotonates glutamic acid, which deprotonates another water, which deprotonates another water. And you could keep going on and on and on and on through this chain till finally you deprotonate one water, which deprotonates an aspartic acid, which deprotonates the last hydronium. So this hydronium right here, this is hydronium number 8. Okay, and this is the hydronium that comes from the matrix. Now, there's several things I want to point out here that we've now generated. Okay, the two things that I've already pointed out is, number one, we've regenerated the copper B in the 1+ oxidation state and the heme A3 iron in the 2+. Okay, so copper B 1+ oxidation state, that's check, and iron in the 2+. That's how we started off this mechanistic reaction. Okay, now also notice that this histidine right here is in the protonated state. If you scroll back to the first step, notice that this histidine was also in the protonated state. See? So that's also something that's important to realize is, number one, we've regenerated histidine in the protonated state. And also, what's also key to understand is the tyrosine is also in the protonated state. We want that to happen. Okay? Now also, the last thing, and probably the most important thing I want to point your attention to, is notice what started this reaction. Okay? Let me do this in, what's a good color? Let me do this in orange, just so you can kind of see it. Number one, what was our original substrate for cytochrome C oxidase? Well, it was molecular oxygen. And what do we get at the very end of this reaction scheme? We get, circle it in orange as well, 2 water molecules. Okay, so if we go back to the original basic reaction of cytochrome C oxidase without the mechanism, I said that we pump 8 protons in from the matrix. That's what we did. And then we pump 4 of them into the inner membrane space. And that's exactly what happened here. And then we had a leftover of 4 protons that didn't get pumped into the inner membrane space. And where did they end up? They ended up on the 2 atoms of molecular oxygen that became ultimately 2 molecules of water. And these 4 electrons right here, these 4 electrons ultimately came from cytochrome C in the reduced state. Or we can call that ferrocytochrome C. Okay? So hopefully this video gave you a little bit of intuition on cytochrome C oxidase. In the next few videos, we're going to go over some more facets of cytochrome C oxidase that might be interesting to you. But hopefully this gave you a little bit of intuition. See you in the next video.